Biology students, we're on 10.3. We're at the end game for chapter 10. And although this is going to be a short section, it's full of a lot of information. So it's all about gene linkage and polyploidy. The crossing over of linked genes is a source of genetic variation. And of course, that should seem very familiar to you back from section 2 when we were talking about Mendel and even prior to meiosis. Genetic variation, we have it for a reason, because why? It would be boring to see everybody look alike. So first we have to kind of discuss what genetic recombination is. The new combination of genes produced by crossing over and independent assortment is just that, genetic recombination. Combination of genes due to independent assortment can be calculated using that formula 2n, where n is the number of chromosome pairs. Any possible male gamete, sperm, can fertilize any possible female gamete, egg, so that the possible combinations after fertilization are 2n crossed by 2n. Genes located close to each other on the same chromosome are said to be linked. They usually travel together during gamete formation. Gene linkage results in an exception to Mendel's law of independent assortment. So in this bubble, you can see replicated homologous chromosomes. And after it goes through the first stage of meiosis, the homologous chromosomes separate. But because it's meiosis, it goes through a second separation during meiosis 2, where centromeres separate and the gametes form. And here we have our 4 different types. Crossing over occurs more frequently between genes that are farther apart. Crossover data can be used to create chromosome maps, which are depictions of how genes are arranged on a chromosome. So here you can see there's a type of fly, right? And we can see that we have a wild type. And this stands for yellow body color, white eye color, vermilion eye color. Now that's going to come in handy later if we do an independent lab. So polyploidy is the occurrence of one or more extra sets of all chromosomes in an organism. A triploid organism is designated 3N, which means that it has 3 complete sets of chromosomes. So when does that happen? Well, many agricultural crops are polyploid: wheat, oats, and sugarcane. Polyploid plants often have increased vigor as well as size.
But we're just adding a little bit more to it with the new syllabus as well too. So speciation means creation of a new species, and this can happen in many different ways, and different scientists will debate about what needs to happen in order for a group to be considered a new species. So they can be separated by geographic isolation or behavioral isolation, certain animals not responding to signals being sent by other animals, and therefore they don't mate under natural circumstances. Then scientists might say, okay, I think they're different enough to call them different species. Well, here's another way. This word polyploidy should sound familiar because you know the words diploid and haploid. Diploid meaning 2n, we'll see up here in a second, and haploid meaning n. We know that gametes are n, and body cells or somatic cells are 2n. The magic numbers for humans are 46 and 23 sperm cells. 23 chromosomes, in a sperm cell match up with 23 chromosomes in an egg cell to produce the zygote, which starts with 46, and then all of our body cells come from that as well too. So looking at the prefix here, which is useful for all kinds of biological terms, if we put the prefix poly into this ploidy, then we end up with a condition where organisms contain more than 2 homologous sets of chromosomes. And this can happen in lots of different types of organisms, including the ones that are pictured right here. If you have 3 copies of each chromosome, it's called triploid. 4 is tetraploid, so on and so forth, pentaploid, hexaploid. And actually, the combining and recombining of all these different combinations of sets of chromosomes, usually through errors, it's not intentional, but through these mitotic errors, will actually produce technically new species. Even though they may only be using the instructions from very few of these chromosomes, but actually the extra copies, which causes the polyploidy, by definition is a way to lead to a new species as well. And you should know that non-disjunction can lead to errors. In humans, obviously non-disjunction, the most famous case of non-disjunction is when the chromosomes don't separate properly during either anaphase 1 or anaphase 2, which results in extra chromosomes ending up in the gametes. And obviously, although 46 is the famous number for humans, some kids can be 47.